Good morning, all of you. So, we are discussing now here Unit 2 Immunological Tolerance and Autoimmunity. In our previous discussion, we studied regarding the hypersensitivity type of reactions, in which we discussed four different types of hypersensitivity reaction, including type 1, type 2, type 3, and type 4. So, everywhere we studied that the mechanism of allergy or mechanism of hypersensitivity reaction is different here. In type 1, we discuss anaphylactic type of reaction in which, or allergic type of reaction, in which IgE production is increased or there is overproduction of IgE antibodies. And these antibodies, they are having ability to bind on the master cell surface, and after, along with the binding with antigen to the master cell surface, there is a degranulation of mast cell releasing pharmacologically mediators, and the effect of this mediator is nothing but allergic response. In type 2 reaction, we studied blood transfusion reaction, in which we studied that IgG or IgM type of antibodies are involved, and blood transfusion reaction, that is whenever there is a transfusion or transfer of blood from one person to another, the proper cross matching of blood group is necessary, otherwise it will create some complications. In type 3 reaction, we studied the immune complex type of reaction in which antigen antibody complex is formed, which is fixing complement, and that leads to certain abnormal reaction, that is Arthur's reaction or serum sickness type of reaction, in which the foreign serum which will introduce in the body, the antibodies are almost foreign to us, and against that there may be response. In type 4, we studied delayed type of reaction, that is example is allograft reaction, in which transplantation process we studied and how there is a response against the alloantigen is developed, and due to which the graft is rejected. So this is regarding the type 4 hypersensitivity and total hypersensitivity reaction in brief. Now we are studying tolerance and autoimmunity or autoimmune response. So what is immunological tolerance? What is the concept of tolerance? So we know that when there is a contact of antigen, the response is developed by our body, and that response is called as immune response, as it is protective to us. Immunological tolerance means a state of unresponsiveness. It is a condition in which there is no any response. No response is developed against the antigen. Means that the antigen will be tolerated by the body, though no response at all. जेव्हा कुठला रिस्पॉन्स निर्माण होत नाही CMI किंवा AMI अँटीजेनच्या अगेन्स्ट, तेव्हा त्या स्टेटला टॉलरन्स म्हणायचं. द टॉलरन्स इज अ स्टेट ऑफ अनरिस्पॉन्सिव्हनेस. हिअर no any type of the response is developed against the antigen. The immune system is tolerance against the self antigen, hence does not show any immunity against the self antigen. Generally, tolerance is developed against the self antigen. Self antigen means those antigens which are present in our certain organs in our body constantly. So against the self antigen, response is not generally not developed. If it is developed, then it will lead to autoimmunity. But generally, the response is developed against the foreign antigen, not against the self antigen. But whenever the response is not developed, it is called as state of unresponsiveness or tolerance. In tolerance state, contact of antigen specifically destroys the capacity to mount an immune response against the antigen. So here, whenever there is a subsequent contact of the antigen, this antigen contact destroys the capacity to mount an immune response. Means either there is no development, or there is no maturation, or you can say no proliferation of T cell or B cell, and therefore there is no any formation of subsets as well as immunity, neither CMI nor AMI. Means what? the antigen destroys the capacity to mount an immune response. Now, what is meant by self-tolerance or what is a natural tolerance? So, natural or self-tolerance against the self-antigen is called as self-tolerance. When the tolerance is developed against the self-antigen, that is called as self-tolerance. When there is no any mounting of immune response against the self-antigen, then it is said to be self-tolerance. So, no response is developed, neither CMI nor AMI, any type of the response is not developed there against the self antigen. So, in that case, it is called as self tolerance or natural tolerance. But it leads to sometime autoimmunity and it is very harmful to individual. If in case, if the response is developed against the self antigen, suppose AMI or CMI, then it is dangerous to us, that is harmful to individual, and that lead to autoimmunity or autoimmune diseases. Autoimmunity means what? Auto means self. Immunity means protection. Protection against the self antigen, that is called as autoimmunity. In generally response is not developed, but in case if response is developed, if immunity is developed against the self antigen, then that will be harmful really to the individual and that lead to certain diseases, that is the autoimmune diseases. So tolerance to self antigen is very significant and as it is applicable in transplantation as well as in controlling autoimmunity. So when the tolerance, why it is necessary to develop tolerance? If tolerance is developed, no response is there. So against the self antigen, when a tolerance is developed, that is very important because it is having application in transplantation. Transplantation reaction means whenever there is a transfer of the certain organs or graft from one individual to another individual. As well as the self tolerance is important in autoimmunity. So if suppose if the tolerance is not there, if immunity is there, then antibodies will be produced against the self antigen and that antigen antibody reaction will occur and it is harmful to the individual. So tolerance is important, tolerance is necessary in that case to control autoimmunity. If tolerance is there, no autoimmunity. व्हॉट इज इंड्यूस टॉलरन्स? इंड्यूस टॉलरन्स म्हणजे काय? आपण निर्माण केलेला किंवा डेव्हलप केलेला. नॅचरल टॉलरन्स म्हणजे सेल्फ अँटिजेनच्या अगेन्स्ट ज्यावेळेला रिस्पॉन्स कुठलाही डेव्हलप होत नाही तेव्हा तो सेल्फ टॉलरन्स. इंड्यूस टॉलरन्स म्हणजे आपल्याला मुद्दामून टॉलरन्स स्टेट निर्माण करायला लागते. So extensive research is focused on immunological tolerance because it finds application in transplantation and in controlling autoimmune diseases. So tolerance state is important because it is having application in transplantation, graft transplantation as well as in regulating or controlling the autoimmunity. Why it is necessary? If suppose the transplantation is there and the graft or the organ which is applied on the individual that is almost foreign to that particular recipient body, and along with that organs some antigens are present along with that organs, so these antigens will also enter in the recipient body. So whenever the organs are transplanted, naturally along with the organs antigens will be also there, and these antigens are foreign to the individual almost because two individuals are genetically different. So the antigen or the organs which is transplanted and whatever alloantigens are there along with that organ, they will be treated as a completely foreign by the recipient body, and against that antigen antibody response will be developed. This is one thing. So if such things should not happen, then we have to develop the tolerance. असं जर व्हायला नको असेल तर टॉलरन्स डेव्हलप करायला पाहिजे. सो इफ टॉलरन्स स्टेट इज इंड्यूस म्हणजे रिस्पॉन्स कुठलाच डेव्हलप होणार नाही ॲलो अँटिजेनच्या अगेन्स्ट म्हणजेच टॉलरन्स स्टेट, तर त्या ठिकाणी ट्रान्सप्लांटेशन सक्सेसफुल होऊ शकतं. सो इफ टॉलरन्स स्टेट इज इंड्यूस टू ॲलो अँटिजेन, इट मे रिझल्ट इन ॲक्सेप्टन्स ऑफ ग्राफ्ट अँड मे कंट्रोल ऑटोइम्यून डिसीज. सो बोथ द थिंग्स हॅपन देअर. They are helping in transplantation or successful transplantation. Side by side, it is also helpful in controlling autoimmune diseases. So this is one significant application you can say regarding the development of tolerance. Now, how tolerance is induced? See here, in neonatal or in small animals, or you can say just newborn animals, it is easy to develop the tolerance. Why? Because the organs are not well developed and they are not get properly matured. So in that case, if we introduce any antigen, then the body of that particular animal is not completely developed. All the immunological systems are not completely well developed. So it is not known whether the antigen is self or foreign. It cannot distinguish the self and foreign antigen. Whatever antigens we are introducing in that particular neonatal stage or in the fetus or a fetal animal, then in that particular condition, the antigen will be treated as a self antigen. No response will be developed because the immune system of the animal body is not matured, is not completely well developed, and therefore it cannot distinguish between self antigen and foreign antigen. And therefore, in that condition, whatever antigens you are introducing in the body, these antigens will be almost treated as a self antigen. No response is developed. So it is easy to develop the tolerance, to induce the tolerance in neonatal animal or in the newborn fetus, newborn animal. But it is difficult side by side to develop the tolerance in adults. Adults means developed animals. So whenever it is necessary to develop the tolerance, to induce the tolerance in young or in adult animals, that is slightly difficult because adult animals, they are having well developed immunological system. All the systems are well matured. It is completely matured, and this immune system, it can distinguish between the self and foreign antigen easily. So it is difficult to develop tolerance in case of adult animals. So the induction of tolerance in adult is important as it is used in transplantation reaction, because we are doing transplantation later or after this, that means after embryonic life, later on whenever there is a problem. But in that case, it is necessary to develop the tolerance because whatever is organs which are to be transplanted, these organs should be successfully transplanted and they should be functionally well in that particular body. But it is possible only when the tolerance is developed, as the organs are having alloantigens, and when the organs are transplanted, the antigens will be treated as a foreign almost, because two individuals are different, genetically different. So even though the purpose is good one, but the anti-immune system of the body is differentiating between the self and foreign. It will be treated as a foreign antigen, though it is a grafted tissue or what it may be. Whatever antigens are coming along with that, these antigens will be treated as a foreign antigen by our immune system, because in adult all the systems are well developed, they are mature one, and they can easily differentiate self and foreign. So it is one problem. So in that case, it is necessary before the transplantation, the tolerance state should be developed so that the transplantation will be successful or the graft can be accepted by the individual body. This is one thing. Now, how the tolerance is then developed in adults? How it is possible to develop the tolerance in adult? Because we know that all the systems are matured and well developed. They are functioning very well in the body. So they are doing their work. Means immunological system doing their work means develop the antibody response against the foreign antigen. Only this thing is known. So how to develop tolerance in case of adult? If suppose we are transplanting the graft, if it is transplantation process, then it is necessary that before the transplantation it should be well that means accepted. It should be accepted by the body. It should be functioning in that body. It is very important. If the organ is transplanted and if it is rejected by the body, there is no use at all of the transplantation process. So before the transplantation, it is necessary to develop the tolerance state in the recipient body in adult. So how it can be induced? So one thing is that the tolerance can be developed in the body by using either monoclonal antibodies or drugs, or by using anti-lymphocytic serum, or by using immunotoxins, or by using immunosuppressive drugs. So by using certain specific monoclonal antibodies, you can develop the tolerance. By using certain drugs, you can develop a tolerance. Immunosuppressive drugs you can use, which can reduce immunity, which can suppress immune response. And anti-lymphocytic sera. Anti-lymphocytic sera means what? the serum containing antibodies against the lymphocytes. So these sera can be used in the person to develop the tolerance. And immunotoxins. Certain immunotoxins are also used to develop tolerance there. So what is the major mechanism here? Means what happens if we are using such type of the drugs, such type of the antibody, such type of the certain type of the sera. So many different methods are given here. So when we are using all these things, what is the major mechanism which happens there? If we are using antibodies, if we are using specific monoclonal antibodies, if we are using anti-lymphocytic serum, then that whatever antibodies are there, they may react or they may be responsible for the inhibition of T cell or B cell growth or proliferation of T and B cell. If there is no any proliferation, inhibition of the proliferation means what? T cell and B cells, whenever they get induced by the antigen, there is a proliferation of T cell, there is a proliferation of B cell. Once the B cell get proliferate, there is a plasma and memory cells, and plasma cells are secreting antibodies. If there is no proliferation at all, then there is no formation of memory cell, no formation of plasma cell, no formation of antibodies at all. It means that there is no antibody response. AMI will not be developed. In case of T cell also, the T cells are thymic cells and which are inducing CMI. We know very well, cell-mediated immunity. So whenever such type of the sera, whenever such type of the antibodies, whenever these drugs we are using, they may interfere in the development or maturation of the T cell and the proliferation of the T cell also. It means that these media in the sense, these drugs, antibodies, whatever we are using, they may interfere in the development, they may interfere in the proliferation of either T cell or B cell. So whatever response is given is developed by T cell or B cell, that is CMI or AMI, this response will not be developed, because as there is no proliferation of T and B cell, there is no formation of the T cell subsets, there is no formation of the plasma and memory cell. So how there is development of CMI and AMI? No development of CMI, no development of antibody, no development of cell-mediated response. It means that neither CMI nor AMI will be developed there, and it means no response at all, means a state of unresponsiveness, that is a tolerance that can be developed. So in order to develop specifically tolerance, either we have to use monoclonal antibodies or we have to use immunosuppressive drugs, methotrexate like drugs we are using. So certain drugs we can use here. See, one example is given here, how tolerance can be induced by monoclonal antibodies. We know monoclonal antibodies these are the antibodies which are very specific against the single antigen determinant. That part we have already discussed in our first unit. So when these antibodies are using, then what these antibodies, what type of the mechanism is there? What is the function of these monoclonal antibodies? These antibodies have specificity against a single antigenic epitope, and these can be used to suppress the activity of T cell population. So what these antibodies are doing when we are going to administer monoclonal antibodies, these monoclonal antibodies, they are going to suppress, they are going to interfere, they are going to reduce the activity of T cell population. T cell population means T cell, they are proliferating into T cell subsets. T cell subset means T helper cell, T suppressor cell, effector T cell, regulator T cell, like that. So whatever proliferation of T cell is there, this proliferation will be inhibited by monoclonal antibodies, as these are very specific towards the single determinant, antigen determinant. So monoclonal antibodies are very specific and therefore we are using it deliberately. So these antibodies, they are interfering the T cell population, activity of T cell population, and therefore no CMI will be developed. This is one thing. Secondly, when we are using immunosuppressive drugs, by using immunosuppressive drugs we can develop the tolerance. Okay, how? What is immunosuppressive drugs? Immunosuppressive drugs means these drugs, they are reducing, they are suppressing the immunity. That is the immunosuppressive जे ड्रग्स इम्युन रिस्पॉन्स डेव्हलप होऊ देत नाहीत त्यांना म्हणायचं इम्युनोसप्रेसिव्ह ड्रग्स. इम्युन रिस्पॉन्स थांबवण्याचा प्रयत्न करतात किंवा इम्युन रिस्पॉन्स डेव्हलप होऊ देत नाहीत त्यांना म्हणायचं इम्युनोसप्रेसिव्ह ड्रग्स. So tolerance can be induced by using immunosuppressive drugs, and these drugs are mainly used to control T cell population. Again, the same thing is here. Either we have to inhibit T cell activity or B cell activity or B cell and T cell proliferation. So here the drugs which are used commonly to develop the tolerance and to suppress the immunity will be cyclophosphamide 1, methotrexate, azathioprine. These are drugs to suppress immunity and to develop the tolerance in adults. So methotrexate as well as azathioprine, cyclophosphamide, these drugs are commonly used to develop the tolerance. So what these drugs are doing, methotrexate, cyclophosphamide, azathioprine, what is the major function of these drugs? So these drugs either control T cell and B cell population or inhibit the proliferation of T and B cell. They are going to control the population of T cell, B cell, or they are going to control the spreading or development or proliferation of T and B cell. If there is no proliferation, the further subsets and plasma and memory cell from B cell will not be produced. As these drugs inhibit T and B cell activity and proliferation, cell-mediated immunity, antibody-mediated immunity will not be developed against the antigen. जर प्रॉलिफरेशन झालं नाही टी सेलचं किंवा बी सेलचं कोणामुळं ह्या ड्रग्जमुळं तर त्या ठिकाणी कुठल्याही प्रकारचा रिस्पॉन्स निर्माण होत नाही. म्हणजे अँटीबॉडीज पण निर्माण होत नाहीत आणि सेल मेडिएटेड इम्युन रिस्पॉन्स पण निर्माण होत नाही. हेन्स म्हणजेच कुठला रिस्पॉन्स निर्माण न होणं म्हणजेच काय निर्माण होणं, टॉलरन्स. स्टेट ऑफ अनरिस्पॉन्सिव्हनेस म्हणजेच टॉलरन्स. कुठल्याच प्रकारचा रिस्पॉन्स निर्माण होत नाही म्हणजेच काय निर्माण होतो, टॉलरन्स. So immunological tolerance can be developed. So if we want to develop the tolerance, we have to suppress the immunity. जर टॉलरन्स आपल्याला पाहिजे असेल तर इम्युन रिस्पॉन्स थांबवायला पाहिजे. जर इम्युन रिस्पॉन्स निर्माण करायचा असेल तर टॉलरन्स थांबवायला पाहिजे. सो दिस थिंग शुड बी दॅट. सो इट मीन्स दॅट टी सेल अँड बी सेल ॲक्टिव्हिटी or T cell and B cell proliferation will be inhibited or suppressed by drugs. कुठले drugs? Methotrexate, cyclophosphamide and azathioprine. So these drugs are commonly used. Other than these drugs, other drugs are also there. But I have given here 3 examples. So these drugs are commonly used to develop the tolerance or to induce the tolerance. So it is necessary to develop the tolerance in transplantation process and autoimmunity control. So why to develop this? It is important in transplantation reaction, as I said earlier, because if the tolerance is there, then only the graft will be accepted, otherwise it will be rejected. So that is one thing. And secondly, in autoimmunity control, that means whenever we want to control autoimmune diseases, then also the tolerance should be developed. So the tolerance will have 2 applications. In autoimmunity disease control and in transplantation reaction. For successful transplantation, tolerance play a very important role. So this is regarding the tolerance trait and induction of the tolerance. In next lecture, we will discuss the that means control or how the tolerance is developed and what is the exact mechanism behind the tolerance trait, central and peripheral mechanism for tolerance. Thank you.